to our program, Bioinformatics for Precision Medicine. Still have not heard about how all of you are doing. Hopefully, everybody's doing good. So I wanted to start with, again, just a reminder, we had our first session for Omics 2020, a program that just started, and we have another program coming up next week, starting next week, which is going to be Genomics in the Virtual Lab. So just to remind you that we have those programs coming up for those of you that are interested in coding or exploring some of the relationships between sequences and structures of proteins, which we will talk about today in a little bit more detail. But if you're interested in doing that for a whole 2 weeks with an expert who knows a lot about the protein data bank and how this is used in research to explore genetic diseases, viral proteins, and even changes in cancer, you're welcome to take a look at that. So let's take a look at how we are doing as a program. I'm going to just switch over to our program page. This is our program page. So I noticed that there has been a little bit of a slowdown in terms of how long it takes to process some of these pipelines, but you probably saw that some of these pipelines got back online, so to say, or we see some of those getting finished up. So I do want to mention, if you don't see a result immediately for your pipeline, especially some of those bigger pipelines, please don't continue to run them over and over and over again just to see if they work. Because what we had over the weekend is, a lot of people submitted pipelines, and it just takes some time for these pipelines to finish up. The more you kind of put them together, the more you will see that they take a longer time for everybody. So just a reminder, this is something that we need to monitor regularly and make sure that we are not just running, pipelines that essentially do the same thing to the same files, and there's not really a reason for them to run. So that's about the pipelines. I also wanted to point out that we have not seen any project submissions yet. So that's another thing that I think we need to think about, as the program is today in its 7th session. This is the time where we should start thinking about the project ideas. Those of you that have some ideas and you're unsure of how to prepare the project, or have some ideas, you will benefit from taking a look at some of the existing projects that we already have shared with you. For example, there was a question from Yuja, where she was asking, is it free for a registered student? How many can we commit at most? Is it an embedded or more tutorial style? And do we have to finish that in a particular time window, or is it totally self-paced? So the idea of a project is to apply some of the analysis tools to some of the data sets that we've discussed in this program, to do it in a short time. So before the program is over, you still have some time for that program. So, it's something that we hope that you will work on independently on your own time. And we've invited you to a number of review sessions that, hopefully kind of give you a good idea of what those independent projects are. But, I want to go over a few aspects of the project itself so that you can take a look at how they are organized. So let's take a look here. Okay, so again, a lot of activity. So I'm happy to see that all of you are following through with the courses primarily, right? And that's, you can see, is the benefit of kind of seeing what everyone is doing is, quickly we can identify what everybody is working on so that we can address any issues that are coming up. Okay, so continuing to speak about those projects, as you remember, the first step is to frame the problem, right? And that's the time when you want to start submitting those ideas to the form that we shared with you. It will help you organize your thoughts. I've seen many types of submissions. People range from very specific and very detailed to very, very general. So for example, when we look at different types of topics that we covered, we talked about cancer, we talked about infectious diseases, we talk about viruses that cause cancer, right? So we are talking about these different topics, and they're very broad. So these examples hopefully are giving you some clarity on the context. Now, your innovation is the approach to these problems that you're able to explain. So you can say, you know, I'm interested in not just liver cancer, but I'm interested in liver cancer in a particular population. Or I want to see the interaction between, you know, inflammation caused by, let's say, alcohol abuse and infection with hepatitis C or B. If we talk about, you know, breast cancer, the ability to diagnose cancer and treat it at an early stage has improved dramatically over the past decade. So maybe there are still unresolved topics in breast cancer that you might want to research. For example, variability of triple negative breast cancer patients and the implications for their treatment strategies, right? So some of that has to do with reviewing other publications and reviewing literature on that topic. You know, obviously, once you kind of frame the problem and start seeing some data sets on NCBI, yesterday we did a review of NCBI data sets with one of our groups, and not always can you find a data set that is, you know, sufficient samples and enough information about where these samples came from. For example, if you're thinking about something related to specific types of patients, for you to really come to that conclusive answer. And sometimes those data sets are not going to be in the format that you would like them to be. For example, they mention some data set in a publication, but you don't really have access to all of the data. You only have some images that they presented in the publication that are results from that analysis. They're not really the data that the analysis was built from. So that's why it's important to, you know, process or have links to raw data, framing the problem, and that's kind of an iterative process. But once you found those and you are starting to process the data, explore the data, and performing in-depth analysis, you know, that is something that's going to take some time. I mentioned the pipeline issue, and the same issue is going to be there for your independent project, right? It's going to take time to process data, but even that is fairly simple compared to really digging through all of the detail that you will find and giving it some biological interpretation. So we have, you know, several projects right now that different students or program participants are working on, and they're seeing that, you know, they don't really have any results. They've done the analysis, they've come up with a general topic, but to get to that biologically significant and clear result is also a substantial challenge. So there is an alternative. We have a couple of these projects. Okay, that went really fast. So we have a couple of these projects like the patient-derived xenograft models that you went through in Transcriptomics 1. We also have this modeling precision, going forward, modeling cancer precision medicine. So some of those projects are included, and these are guided projects, right? So the first option is you take a project completely on your own, or potentially with another collaborator / participant of this program, and you explore that data set, you know, guided by your understanding of what to do and how to interpret the results. Maybe you're not ready for that. So maybe if you're not ready for that, you can do one of these projects, and what it will give you is an ability to follow through the specific data set, explore some additional questions that were not covered in the transcriptomics courses, and then potentially add some addition, some variation at the end. For example, from all the different subtypes of breast cancer and all the different drugs, explore just one of the drugs and its pathway, and you can maybe identify why that drug will be or will not be effective for a particular breast cancer subtype. Okay, so those are some examples. So before we move on, I just really want to make sure that I answer all the questions that you might have about project submission. So let me start by this. How many of you are actually planning to work on an independent project? So if you are planning to work on an independent project, maybe you can just type in yes in the chat so I will know. If you're not planning to do that, maybe you can type in no. If you're awake, okay, great. Okay, so at least a couple of you. Okay. Okay, so if you're still not awake, then type in coffee. Okay, so the people that are not typing anything, I'm assuming, they're just listening in. So, the reason I'm asking this is because developing your own project and actually getting it to the final result will take some time, and it requires effort on both your part and also appropriate support from our side. So to facilitate that, it seems like maybe there's just a couple of people here that really want to get to that point. As far as I understand, Andrew, you're in another program also. So if you are a part of another program, you can do that there. If you're in this program and you want to finalize a project before the end of this program, I recommend submitting that proposal soon on the form so that we can start understanding, what is your project idea and how do you plan to address the question that you've come up with. Okay, so last time we talked about next generation sequencing in application to viral genomes in the host transcriptome. We discussed how many viruses cause inflammation and variation that leads to cancer. And we have some of these prominent examples here: Epstein-Barr virus, human papillomavirus, and hepatitis B and C. And we looked at one of them, which was hepatitis B and C. We saw that there's variability of the number of infections, and that variability in the number of infections potentially could be linked to some of the prevalence of cancer that we see in different countries. And what's interesting is that we can actually explore whether we can take some samples from, as you remember, SRA, right, Sequence Read Archive on NCBI, and see how there's a difference in the type of genome that we can find in the host samples, right? So if we take the full transcriptome right here, samples, we have here 2 samples, and we process them using HiSat. We use HT-seq to quantify expression. So we can, for example, look at the differences between these 2 samples in terms of expression, which is not going to be statistically significant for anything, right? So instead, we can take the unmapped reads produced by this pipeline and then take the hepatitis B virus genome and do visualization of that genome. So if you remember, the result from that analysis shows us that it's the coverage sometimes, but that's not the full story. There's also an aspect of which elements in the genome are covered by these reads, which gives us some indication into what type of activity that virus might be involved in. For example, is it producing some of these important viral proteins that are critical for it to replicate and to create a virion. So those are some of the additional downstream questions, right? So again, this is just 2 samples or maybe 3 samples that we can pick from a full project. Obviously, this kind of an analysis is not conclusive, right? This could be a random variation. but it is an example of our ability to find viral genomes in the host transcriptome, and our ability to try to link what we see in the data with the biological interpretation. Okay, so has anyone run this pipeline independently, and do you have any questions about what you're finding, or maybe you have tried running this on some other samples? So are there any questions about what we did last time? Okay, if you have any questions, think of them, right? So we have enough time to dedicate to discussing these topics, because ultimately, hopefully this will help you apply some of these tools to a data set of your own. So in this session, we want to talk about multiple sequence alignment and phylogeny. So multiple sequence alignment is the alignment of genomic data. Let's assume some of these genomes that we looked at previously. In this example, we do have some coverage, right, throughout the whole genome. So from this coverage, we can actually create a consensus. And that consensus will represent the full genomic sequence that we see in our data set. Now, we will see also a lot of variation, right? We looked at those particular points where we see a change from the reference genome. So what are these changes? These changes are essentially mutations. So they're variants in genomes, and specific genome, specific types of variations that we find in these genomes eventually lead us to the question, how are they related, right? So first of all, we can characterize the specific mutations by understanding what is their significance or why they occurred. And then we can build a model that can help us study relationships between these different genomes. And that is what is called phylogeny. A phylogenetic tree represents the relationships between the genomes that we find in a sample. So first of all, how can we view these mutations? So mutations, we typically think of them in terms of DNA. As you remember, in DNA, we have base pairs. So base pairs are pairs of bases or nucleotides connected together in the double-strand DNA, and they form tricodons, right? So 3 of these nucleotides form different amino acids. Why do they form amino acids? Because the gene in the DNA codes for RNA, and the RNA codons represent amino acids that will later on fold and create a protein. So when we talk about DNA, we know that DNA is transcribed into mRNA and translated into protein. Sometimes we're talking about viruses that are RNA viruses. So why do you think we still look at DNA sequences when we talk about RNA viruses? Why do you think we are speaking about DNA variation and its impact on the translation of that DNA or the mRNA when we are, speaking about many RNA viruses? For example, hepatitis is an RNA virus. So how come we're speaking about DNA variation? Okay, so the reason is because even if it's an RNA virus, this is called the central dogma of molecular biology for a reason. The central dogma of molecular biology is central to molecular biology because if an amino acid that will form a protein is to be produced, it will follow the same process. And this process is present in all of the cells. So for example, in the human cell, that is what all of the machinery in the cell is built around. And whenever an RNA virus is inside the human cell, it's going to replicate. It is going to trick the cell to think that its RNA is actually the RNA to be in this process. So this process is central, and so we also, that's one side. The other side is that whenever we do sequencing, the sequencing actually is the process of taking RNA or DNA and converting it to cDNA, and that cDNA is actually sequenced. The reason is because DNA is more stable, and so we have much higher confidence in the specific nucleotides that are detected. But these nucleotides, as I mentioned, form codons. And the codons essentially are the building blocks of amino acids. Amino acids are built from trinucleotides. So 3 nucleotides together form 1 amino acid. And so if you look at this example, right here you have 3 nucleotides, here you have 1 amino acid, right? So the sequence in nucleotides is going to be much longer than the sequence in amino acids. And the process of converting one to the other is called codon mapping. And we know that there are specific types of conversions from the different types of changes that we observe in those trinucleotide sequences. Now, these mutations are known to vary between viruses. So for example, here you can see different types of viruses: single-strand positive single-strand RNA viruses, negative single-strand RNA viruses, and so forth. So you have different types. And so we know that DNA viruses are going to have less mutations than RNA viruses. Double-strand RNA or double-strand DNA and single-strand DNA, you can see, are lower than single-strand. And we know that typically the length of the genome is going to be negatively correlated with the number of mutations per replication. So this mutation rate is nucleotide per replication. So when we look at these viruses and study the variation that we observe in data, one of the things that we can do is we can align multiple genomes that we have. So here you can see a nucleotide sequence. Here you can see an amino acid sequence, and they are these are the same samples. But you can see that variation is very different. At the nucleotide level, variation is fairly clear, right? We have here a number of samples and here a number of samples. The separation between them is fairly straightforward. We can observe it right here. But if you look down here, you can start seeing that the actual difference in the amino acid is much, much different, right? So the positions in nucleotides convert to amino acids. Changes in specific nucleotides, which is a mutation, is a change at the nucleotide level, will cause different results in at the amino acid level. And by aligning them together, we can start differentiating between groups of samples and trying to link it to the underlying biology of why that might happen. So why could a mutation occur? So mutation obviously occurs as a result of replication, but let's think about what that replication or adaptation, correct, how is it going to be taking place? So one first example is transmission, right? So transmission between hosts, whether they are the same type of host or different hosts. For example, transmission between humans, or transmission between bats, or transmission between cats, right? Each individual has a different level of immune response, but specifically when that jumps to a different host, whether that is a human or a different type of an animal, the differences in adaptation to the host response, adaptation to the specific receptors that host has, right, that is the strategy for the virus to adapt to that new environment. Sometimes we have intermediate hosts, and the different receptors, right, as the virus tries to enter the cell, it enters through receptors. Those receptors are different between different hosts, and we need to link what we observe in data in terms of mutations, trying to understand how can we describe that process. There's also the immune response and the virus's evasion from the immune response. The only thing that it can do is try to change its own strategy at the nucleotide level. And because of that, we see how this immune pressure is going to be related. For example, viruses in hosts that have strong immune systems against those viruses, they don't essentially get sick. What do? They have a very high pressure on the virus, and they limit its ability to replicate. So when its ability to replicate is limited, and all of a sudden it is in a high pressure environment, it's going to try to change, right, how it evades that suppression to maintain a population, which is its replication process. Now, another aspect is when we observe that adaptation and transmission, we will observe its ability to change specific variation at the protein level, right? Because proteins are the ones that connect to the receptors, and they're the ones that evade the detection from the immune system, for example, right? So because antibodies have very specific sequences that help them recognize these different proteins. So overall, the process that we observe at the nucleotide and the amino acid level is governed by these multiple processes: transmission, evolution, adaptation, and also some level of stochastic process, because ultimately the virus's ability to encode the same exact sequence that its origin sequence had is linked to the fidelity of its polymerase. And so as RNA viruses' polymerases actually are not of high fidelity, those mutations also occur kind of as errors, so by mistake, and we call that a stochastic process. So the ability for us to study the relationship between these genomic sequences and understand how they are related is trying to map how these changes of adaptation, evasion of the immune system, transmission between different hosts, can help us understand the populations that we see in terms of ancestors and present-day species. And then we can try to link the process that we observe at the mutation level to some kind of an evolutionary time, a timeline that explains when that might have occurred. So when we talk about phylogenetic analysis, the input to a phylogenetic analysis is multiple sequence alignment, and the output is a tree that could look like this, or like that. They represent the same thing, which is a relationship between present-day species and their ancestors. So let's take a look at a couple examples. So the first example is a relationship between different Ebola viruses. So here you can see Ebola viruses have 5 different species. Marburg virus is a separate virus that is closely related to Ebola virus, but it's different. You can see here Zaire Ebola virus, Bundibugyo, Ivory Coast, Sudan, and Reston. So these are the 5. And these are a part of the Ebola virus family. So here you can see also inside each one the year when it was detected. So for example, Zaire Ebola virus, right, from 1976 all the way to 2003. Sudan Ebola virus, 1976, similar to this one, and all the way to 2000, 1996, and 2008. So this was before the recent outbreaks of Ebola virus in the West Africa. So when we look at these differences, you can also see that this is the length of 100 steps. So the process of replication happens, and we can see how they are interrelated. So we know, for example, that all of the Ebola virus current species had some origin that is common to them, right? Okay, and if you want to learn about Ebola viruses, by the way, here's a link to a very interesting YouTube video that kind of gives you an overview of some of these things. So how do we actually understand how this tree was built? Okay, let's go to NCBI virus, and let's try to find the first available sequence of an assembled genome for Ebola virus from a human host. Okay, so think of this as an exercise to help you find specific sequences on NCBI virus. So what we need to do is go to NCBI virus. Okay, so NCBI virus. This is the link. Okay, let me know once you've reached this page. So maybe type in yes when you're here. All right, great. So once you're here, let's go to search by virus. I'm going to do this for Ebola virus. You can do it for any other kind of virus for now. So if you just type in Ebola, give it a couple of seconds, and you can see Ebola virus pop up. You can also start recognizing some of these Ebola viruses that we talked about. So let's click on Ebola virus. Okay, and here you can see 3,153 different sequences based on their accession number. But what you'll also see is that these are actually very different, right? So for example, you can see the release date, 2018, 2004, 1999, right? So very different dates. You can see different species, the length of these sequences, right? For example, here we have 483, as opposed to here, 19,724. You'll also see that they come from very different hosts, right? And so forth. So you can actually go here and see some more columns down on the right. So the question was, what is the first full genome sequence from the human host, extracted from the human host? Okay, so using filters right here, you can see different filters. For example, the first filter that we put together is to only look at Ebola viruses. If we clear this, you can see that there's 3,399,000, right? So here we did our first filter, Ebola virus. Okay, now, as you remember, our next question is, what is the first one of the full genome from the human host, right? So we can obviously go to the host and type in human. Choose whichever one you want. So now we're down to 2,400. And also, we're saying that only the full genome. So let's go to nucleotide completeness and just select complete nucleotides. Okay, 446. Now, my question to you. Okay, I'm going to give you a few minutes here to figure out which one was the first one that was that is available from the human host of the Ebola virus. Remember, you can scroll through, okay, and find multiple columns in this table. Okay, so let's see who can be the first one to find the earliest sequence. If you have any questions, you can type them in the chat. All right, we have the first person. Morales is saying 1976. Does anyone else have a number, a year? Okay, Morales is even giving us the nucleotide accession ID. So anyone else? Okay, well, let's see how Morales was able to find that, right? So we can go to collection date. And indeed, we have 1976. Okay, and here he's saying MK114, 118. Okay, so maybe there's more than one. Okay. Okay, so here we see, right, the collection date is what gives us a sense. You can see that some collection date is going to include month and day. Some collection is just going to be year. For some, isolation source, etc. Right? So there's a variation. So the first one, right, obviously 1976 1001 is probably earlier than 1117. So I would say that it is this one. And you can see that it was only released in 2015, right? So in any case, what the point of this exercise is to show you that you have a number of filters and a number of data that you can find right here. So let's take a look, right? If you click on this accession number, it will open up right here, and you can read a little bit more about each one of these. What you can do is select a number of them. So for example, we can select some of these earlier ones, right, from 1976. So let's just select a few. I selected the first 5. Okay, then as you scroll down, let's see, these are 2000, in the 2000s, so 2000. So let's select these 5. And let's take a look at some of the more recent ones. Okay, so here we have 2014. So just select, oops, sorry. Just select, I have to do that again. Okay, so select 1, 2, 3, 4, 5. It's just not reported. So, it's probably in there in the project somewhere, but submission here is only taken from the actual FASTA file, right? So if it's not in the FASTA file, you're not going to find it. Okay, so let's take a few from 1976, a few from 2000, and then scroll down, and let's find a few from 2014. Okay, so here we have 1, 2, 3, 4, 5. So, okay, did everyone select a number of sequences? Maybe put in yes if you have. Okay, I see 2 that have. So let's wait until we have at least 5. So please select, 3 groups: early, medium, and late. So 1, 2, 3, 4. We need one more person. Did anyone else select? Looking for another yes. Going once. Going twice. Okay, sold. So let's go. I see that, not everybody maybe is following along, but let's go and look over here at this button called Align. So you can just click and open it in a different tab. You'll see that it's processing, loading, and here we have multiple alignment. Okay, just one of the several possible ways to do this. Okay, so as you look at this image, remember that what you have in terms of coloring is that it's showing you differences in those sequences. Okay, here you have those those accession numbers that we looked at before, right? And it looks like the higher or the more red the color, the more dissimilar it is between these different strains. So what might be your conclusions about the difference between the very early ones from 1976 and the rest? So let's first ask that question, okay? So what do you think can be obvious from this alignment as you explore what are these different sequences? All right, so how do we know that? We can look here at each one of these accession numbers, right? And so for example, this one, 1976, right? What's obvious is that all of these, not only the plan collection, so not only are these earlier samples, also if you look right here, all of these are from Sudan, so geographically they're from a different location as opposed to all of these are from Zaire, right? So maybe the main factor is not time, right? So maybe the main factor is not the year where it was collected, but actually the location where it was collected, right? The most differences are related to location. Okay, so that is what we can see from this type of an alignment. I think you can also start seeing some additional information, right? So for example, here you can clearly see a couple that stand out. Here you can see 1, 2, 3, 4 that stand out based on a particular difference that we observe here, right? As you look through, you can start identifying specific trends. But how can this tell us which one came from who and how are they interrelated? So now we can go back to NCBI virus, and instead of alignment, we can build a phylogenetic tree. So let's open this in another tab. Okay, so here we actually get a lot more insight about what's going on in terms of the different years, the place, right? And how they are interrelated. For example, we see here a number of sequences, 1, 2, 3, 4, right? So remember when we were looking over here, right? We said 1, 2, 3, 4, okay? These seem to form a group based on this sequence right here. So we indeed see them all from Sierra Leone, and they're all 2014. Then we have 2 from Gabon that are from 2002 and 2001. So let's find those, KC242 and KY471. So 242-800, 242-800, okay, and 242-800, we found that one, and 471-124. 471-124, okay, so this one and this one. If we look at them, right, we see some pattern as well. Here are some red spots. Here you have some red spots. So you see how it's related to the sequence when you are looking at it here and here, right? So here we can see these 4, we can see these 2. And then we have all of these from 1976, but from different locations. Zaire, Democratic Republic of the Congo, 3 of them, etc. So when you look at this phylogenetic tree, right, we can see how it helps us interpret what we see in this page from multiple sequence alignment. And you can play around at different types of visualizations to help you understand better some of the differences. But ultimately, this information can be organized in such a way that it reveals common patterns within groups. The more related they are, the more similar their sequences are going to be. And the more dissimilar their sequences are, the more distant they will be. For example, these sequences are much more dissimilar from the rest of these. And so these form a clade, and these form a clade. Okay? So does all this make sense? Is everybody following along? All right, excellent. So here we have, right, different strains. Let's think about what are the implications. So in the recent outbreak of Ebola virus in 2014, 2016, we know that the virus followed, the spread of the virus followed two separate trends. The first trend is where it potentially originated from. So it originated from different types of bats, and they are considered the possible reservoirs. Then it transferred over to a human, potentially, right, all this hypothetical, from an intermediate host, a gorilla, a chimp, some kind of a wild animal, most likely transferred that virus to the human. And once that virus started spreading in the human population, it also spread in these different countries. So you can actually see this is Liberia, this is Gabon, right? So now we can map what we see in the annotation with the specific sequences that we can find on NCBI virus. So if we take those sequences, we can actually take a look at how they are related. And so if we download here, I have a list of accession numbers that we can download and prepare a phylogenetic tree and build a model, right? So let's do that together. So instead of going to the NCBI virus and just, you know, filtering through, I'm going to give you a specific list of accession numbers. And I'm going to show you how to find these accession numbers, either on NCBI virus or how to find them on what's called batch and trace. Okay, so the two ways that you can do it. The first one, you can just go over here and click on accession and just paste those IDs that I gave you in the chat. Okay, so you can see that we have 9 nucleotides. So please let me know once you've got these 9 nucleotides. It's something that, you know, later on you can use to select other viral sequences if you wish. Okay, so let me know once you've found these sequences, you have them filtered out. Okay, all right, so let's take a look at these sequences. They are from Gabon, 3 from Liberia, 3 from United Kingdom. Okay, different locations. Let's scroll over. You can see these are from 1996, and the rest are from 2014. One is from 2015, right? So we have, again, the same variation. Variation in location, where they were collected, and variation in time frame. Okay, now you can click on download. Here you can see that you have nucleotide, and that's what we want. We want the FASTA files from nucleotide. So click on next. Okay, and we have all our records. Here you can do use default. Okay, so what did we do? We generated a FASTA file. Okay, let me know if you were able to generate the same FASTA file. Okay, great. Let's take a look at this FASTA file. So you see the accession number, the description, right? Where it came from, the year, etc. How many do I have in this FASTA file? If I do Command F, just search for, right? If you search for this symbol, okay, you can see that you have 9 of them. So this is just a way to check. So we have 9 different sequences, one after the next. One sequence finishes, another one starts, right? And they're organized like this. So now let's go to the server platform, right? And we'll kind of understand how this could be done. And here we have this section called phylogenetic tree. Okay, so you have, let's, by the way, change the name. So we'll call this Ebola virus. What is another thing that you might need for your analysis? Another thing that you need is a reference genome, right? So how do we find the reference genome? Here, you can go to sequence type and find the ref-seq, okay? And you have these different reference genomes. let's take the Zaire Ebola virus. So if you click on this reference genome, let's take a look. This is a complete genome, right? How do we download this? We actually go to send to, file, and then download it as GenBank. Okay, create file. Okay, so if there are any questions or you want me to pause, let me know. Okay, so now we are ready. We have our Ebola virus of multiple types of Ebola viruses. We have the reference genome. So reference Ebola virus Zaire. Okay, is everybody with me? Okay, so here we have FASTA, change it from FASTQ to FASTA. And now let's upload these files. So here, first of all, we'll do upload file. So we'll add that file. The file we want to add are the different Ebola viruses that we downloaded from NCBI virus, right? Okay, so here it is. Let's go to group selection. Do we have any groups? We don't, right? So let's just add it in here. Okay, now we need the GenBank file for framing codons. Remember what those codons are, right? So here we have this GenBank file. So why, by the way, what type of information does this provide us with? Reading frames, right, depends on where you start. You can start with 3 nucleotide sequences from the first, but you don't know where the first one is. Oh, okay. So where do we go on the server? So this is the server. You go right here, phylogenetic tree. Okay. All right. So you go in here, and then we uploaded the files. Here you change to FASTA, and then you add your files right here. So that's what we did up until now. Then we uploaded 1 GenBank file for framing codons. There's an option for us to include some sequences with a particular time. We already know the time, so we don't need that. So we can go to, okay, we can go to skip. So we have a reference, we use it for codon mapping, and we have the FASTA file, okay? So if you're here, now you click on start. We have a section here for multiple sequence alignment of FASTA files, right? So multiple sequence alignment, what we did on NCBI. We can convert that to amino acid sequences, and we know that different changes at the nucleotide level will impact differently the different amino acid sequences. And then also, in some cases, the evolutionary process, right? Remember, what we're trying to study here is a biological process by looking at sequences. So the more we can incorporate our knowledge of the biological side of evolution, the more we will get an accurate result. And here we are taking this specific reference that talks about strong purifying selection at synonymous sites in D. melanogaster. So this is a study that shows that the positions inside a codon that actually corresponds to evolution, to this adaptation, are not random. It's not just any nucleotide in a codon. Some specific codons actually have a greater representation of evolutionary relationship than if we just assume that they are random. So here, we don't really need to incorporate it here right now. So let's just keep it as false for both. And now you see that actually there are many different types of phylogenetic analyses. Some are based on clustering, sort of, 8 here is the closest to hierarchical clustering that you might be familiar with. It just looks at similarity in this multidimensional space and helps them organize into groups. Very similar to what we saw on NCBI. Some additional models that you see here are called BEAST. So BEAST is actually a process of trying to, again, incorporate some of this evolutionary analysis and statistical analysis to try to represent the biology of what's going on inside this population in a better way. So for example, we have multiple models here. Coalescent, that assumes an unknown constant population size back through time. Okay, or let's take a look at another one. This process assumes an unknown constant lineage birth rate for each branch in the tree. This one is based on the birth-death model where there are both birth rate and death rate present, right? So as you select some of these, you get different ideas about, you know, how they look at the population that we assume co-evolved and has led to the sequences that we observe in our data set. Okay, so you also see there some option to change it from nucleotide to amino acid, right? Depending on what you are more interested in. Are you interested in all of your mutations or only the mutations that lead to changes in amino acids? Because not all of them will do that. So let's select birth death for now and keep it at nucleotide. Click on save and end. Okay, and we'll call this Ebola virus evolutionary beast. Okay, run pipeline. So if I can get a sense of how many of you started a pipeline, maybe you can type in pipeline for me to know how many of you actually started this. All right, so Mani, anybody else? Did anyone else start this pipeline? Did anyone else have a chance to start, or do I need to go through it again? So remember, you can choose different models, right? So right here is the pipeline that we built. We started from here, went through multiple sequence alignment, converted to amino acids, looked at specific positions in the codon, then selected BEAST as, well, we selected birth-death, but this, yeah, birth-death process, and then end. And remember that we have a number of different variations here. Okay, so as you wait for your pipeline to finish, let's take a look at the output. So I already previously ran this pipeline. And here we have some of the separation built by this model. So the model shows us that here are the later Great Britain. Here are the later Liberia. And a completely different clade is around this earlier sequencing from Gabon. So we can see that the relationship between these sequences is very closely related to these different sequences that we selected. But there's more to the story. In fact, if you look at the type of data, if you look more closely into how this data was generated, you'll find that some of these were created by sequencing directly from the wild type or directly from patients. And in other cases, it was done through tissue culture, right? So there's a lot more to the story if you actually start digging through the details of how the data was generated. And that might explain some of the differences that you can see. But what's important for us is that we can see that this type of analysis, phylogenetic analysis, shows us that this is a much earlier, right? You can see that this clade is much shorter because the distance here is evolutionary time, right? So these have originated from some common ancestor not so long ago. As opposed to these, this was farther away, and potentially you can see here that we used the reference and the early time sample, right, to represent time more accurately. And you can see that this is more stretched out. You can see that they're also separated significantly between themselves. So we know that there's a fairly large difference between them. Let's take a look quickly at another example. So all of us know that there's a big difference between these different types of respiratory viruses, flu, COVID-19, and the previous coronaviruses like SARS and MERS. So for example, you can see that SARS happened in around 2003, MERS happened around 2014, and the COVID-19 obviously is ongoing. So what's different between them? The reproductive number is different. We know that the case fatality rate is different, right? So they produce different phenotypes, different diseases, different pathogenesis, etc. We can actually observe the spread of the virus in almost near real time by looking at this phylogenetic tree. So let's take a look at that phylogenetic tree together. Here's a link. Paste it in chat. Okay, let's go there quickly. This is a project that is based on sequences submitted in near real time through GIS-8. Okay, an international project, and here you can see our phylogenetic tree. But instead of just a phylogenetic tree, we have some additional information. The additional information are the locations of these viral genes that are collected in different countries. And here you can see also the variation in the coronavirus genome as it spreads. So what you can do here is play this information, to see how it spread over time. So let's do that. Okay, you can see that as it started spreading from China, it started separating into these major clades, right? So here's the blue clade. Here's the green clade. Then we started seeing how the blue clade is the one that went over to North America, right, primarily. And the yellow one went to Europe, and then they started interchanging, right? So potentially Europeans came to the US as well. So you can start tracking, right, how the virus, based on its sequence, spread throughout the world and started separating into these different subgroups, linking that to location of more stable populations of patients, of humans. And of course, at some point, the lockdowns happened, right? You can see here the individual months, and you can see how these clades became more formalized. And we can even see, based on the multiple sequence alignment, right, we can start asking additional questions, not just whether they are different or how closely or farther away they are positioned in our evolutionary time, but we can also look at the specific positions where the most pronounced differences can be found. So here you can see the genome, right, and the genome is from 0 to about 30,000 nucleotides long, and you can see the specific positions where there are very prominent mutations. For example, if you hover over here, you can see that this is codon 614 in protein S. Okay, remember that number, 614 and protein S. So let's take a look at how we can do a similar analysis. Again, we can go to all of these coronavirus strains. You can see if you just type in Coronaviridae, which is the coronavirus family, you can start seeing how we can find different strains, different countries, different hosts, different country of origin, GenBank accession, and we can start grouping them into a number of different groups. For example, an analysis that we can do is take a number of equal number of sequences from different strains of coronavirus, including SARS, MERS, COVID-19, beta coronaviruses, analyze it in the very same way. You can see here a different section, but the same steps, right? Multiple sequence alignment AA, codon MSA, and then we did all phylogeny methods and next tree. What do we see? We see a fairly clear separation into defined groups. Another type of a comparison that we can do is here comparing the original SARS. It happened in 2003, but it continued to affect some people. So the question is it just an evolution, a natural evolution over time of the original SARS sequences, SARS viruses, or is it some kind of a novel strain? And here we can see that it is very clearly a novel strain, right? So these sequences from 2004 and 2017 are related to each other, fairly distant from each other, but very different from these novel coronaviruses that we see down here. We can also see that this particular group is very different from the rest of them. And this is just US and Europe, right? Not China, right? So we know that there is a group of samples that probably are linked to European origin as opposed to directly from China, right? And there's actually evidence for that because some of the patients traveling to the West Coast originally came directly from China. Some of them came from Europe, right? So those types of things help us trace how we can study the origins of this novel coronavirus as it spreads in the population. As I mentioned, we can also look at how these different sequences can be organized into groups, right? So what is specific to these groups? What is specific to these groups? What is specific to them are specific mutations. So here you can see now a circular phylogenetic tree and the different clades of the phylogenetic tree, the G clade, S, and V, and then other. So here you can see that these three strains could be defined by specific variations, mutations. You remember the number 614? It's right here, G, right? So this whole group right here has a change in the protein S. In position 614, it's a change from D to G, right? And the same is true for these other ones. But over time, as more data was collected, those started breaking down into more. For example, the G started becoming GH, G, GR, S, V, and L, and other, right? So now we know that the coronavirus is actually very different as it evolves through this population, but it does not mutate everywhere randomly. It actually mutates in very specific positions. How do we know what is the impact of those positions? Well, we can do the same analysis. Here you can see two examples. One example is here. You can see the difference between different hosts, so bats, pangolins, and humans. And here you can see that mutation 614 from D to G. You can see some sequences, right? This is the spike glycoprotein, a specific region, which is close to the cleavage of the spike glycoprotein, starts to be changing from D to G, right? So that gives us a sense, because we know what these amino acids are and what are their physicochemical properties, we can start understanding the impact on the physical or the physicochemical interaction with other proteins. Let's take a look at what those might be. So we developed this visualization tool to help us explain and visualize some of these differences. It is called SeqNav. So this is the link. Okay, so here you can see different data sets, different comparisons between these different viruses. For example, here you can see a collection of different genomes that we were just looking at: MERS, SARS, COVID-19, and beta coronaviruses. Okay, again, you have the same kind of a difference explained in colors. So the more red, the more difference you will find. This group right here has all gray, right? Why is that? Because the reference is actually COVID-19. Okay, so if you actually look at this reference, this is COVID-19. this is COVID-19, right? And then you can see that which one do you think is the more closely related strain to this novel coronavirus based on what you see here? Okay, so what group does it belong to? SARS, exactly. So SARS-1 is the closest to COVID-19, right? And typically, you know, these variations within the group is what we're trying to avoid, right? We are trying to represent groups by a collection of samples so that we have a better sense of what is the whole group like. So you can now click here and kind of see where it's from. So the important thing, though, is that here you can see also the proteins of this virus. So we talked about this 614, right? So you can click on the surface glycoprotein. The surface glycoprotein is what connects to the host receptor. You can see that it is right here on top, has a receptor binding domain. And this receptor binding domain is actually important to its function. So right here, right around here, right, you will find some of those, right? So if you hover over, you can see the numbers, right? These are the numbers of amino acids. Okay, so this is a specific example, right, of how we can link the sequence data to the structural data. And the significance of that is that it can actually explain a number of things that we might be asking about. For example, here you can see the difference between the bat coronavirus closest genome to the human COVID-19, just looking at the amino acid sequence of the two spike glycoproteins. And if we map those, you can see that they are positioned not by chance, right? They are positioned in a specific region. We know that this region is actually responsible for the interaction between the viral protein and the ACE protein on the surface of the receptor of the cell, the host cell. You can do the same thing, the same phylogenetic analysis on the ACE2 receptor. So the ACE2 receptor here is compared between different sequences from humans, right here, and other types of potential hosts. Okay, so you can see this is a bat, large flying fox. This is a Chinese rufous horseshoe bat. Here's pangolin. Here's house mouse. Southern multi-mammoth mouse, gray mouse lemur, and 10-building bat, right? So some of these bats are not a problem. Some of these bats are not a problem. But some of them, right, now we can see that, for example, house mouse, right, is not that far away from the human. And so now we can think about different types of intermediate hosts that might be an issue when we think about this coronavirus spreading in the population. And the specific position, so you can see here the interaction between, this is the human SARS coronavirus receptor binding domain, and here's the human ACE2. Here's the interaction between them, right? The interaction between them is actually dependent on only a small number of amino acids. So how the virus spreads, remember we looked at an overall influence of different processes, evolution, replication, transmission, and some of those things could be studied at the level of phylogenetic relationship and multiple sequence alignment to demonstrate how this can actually be of potential risk or significance as we think about this coronavirus infection. Okay, so any questions about what we covered today? Okay, what is the question? Step 5. Okay, so let's go quickly through the pipeline. Okay, so we go to phylogenetic tree. And stop me if you have a question. Here we select FASTA files. Okay, and go to upload the files. So uploading files is right here, add files. So let's call this step number 3. We select the FASTA sequence, which we can have just one or we can have many. For example, we can organize them by group if we want to. So we can then select groups and drag them in here. Right? So we could have multiple groups, but we don't, we just have one file. Okay, what is framing? Framing, as you remember, each nucleotide sequence can be converted to amino acid sequence, but it depends which one you start from. So this GenBank file contains both. It has the nucleotide sequence and it has the amino acid sequence, giving us a clear example of how to associate the two sequences. So here, we need to add the GenBank file, GB, and not the FASTA. Okay, we have the option to add some files that are outgroup, but we don't need them. So we can just click on skip. And now we can build the pipeline. What are the steps? We start, select multiple sequence alignment. This is using Clustal W, version 2.0. We theoretically could end here, but instead we want to do some of this analysis, and we could do it only on nucleotide sequences, or we can convert it to amino acid sequences and incorporate some evolutionary dynamics that have been shown to be significant for evolution, which is the position in the codon. And now we can select multiple types of phylogenetic trees, or just click on all phylogeny methods. Here you can choose between amino acid and nucleotide sequences. Tree space allows us to study the different trees and come up with the best representative trees of what we see in our sequence. Okay, and now we can click on end. Okay, did you have any questions there, Morales? All right. Okay, any other questions? Yes, please. Okay, so what do you mean by circular genomes? Yeah, so any viral genome that you will find will come in a FASTA file, right? So if you find that genome, any genome here will have a FASTA file. Whether in real life it is a circular genome or not a circular genome, does not matter for alignment. Yes, it's not supposed to be gb.txt, it is supposed to be .gb. Okay, so you look right here, it is .gb, not gb.txt. So how do you download the reference genome? So for example, this genome right here, okay, I can send to file, and here select format GenBank. Okay, what is the GenBank format? The GenBank format is different from the FASTA format because it contains annotation. If I open this up, you can see all of this annotation, right? So this is your GenBank file. This is your FASTA file. And you can see here, this is translation. So this is translated into amino acid sequences. And at the bottom, you have the nucleotide sequences. Okay, Sarah? in the case when the NCBI, we had some virus that began in different parts of the genome. Well, it doesn't really matter where it begins. Well, here, if you select GenBank, right, and do create file, it downloads it as GB. Now, if it happens, for some reason, your computer is changing it to TXT, just change it back to whatever that GB. Okay, so Morales, like I said, viruses, if we have a known sequence, that sequence is going to come as a FASTA file, right? It will come as this. If you have that FASTA file, it doesn't matter how that file looks like in real life. What matters are the differences in sequence from the reference, right? Now, there might be some additional considerations. I actually don't know a lot about circular stranded genomes, viruses. You might know a lot more than me about that. So I haven't really looked into what issues are going to be, if you do that kind of alignment. But I don't see how that will matter because the sequence should be aligned. And then, potentially the different starting regions is an additional step that you might want to do for that specific type of viruses. I don't know. Okay, so we talked about these, the phylogenetic analysis, right? And let's kind of review where we are. We met today to speak about multiple sequence alignment and phylogeny. We started a pipeline. And you can start a different pipeline, right, with your own sets of viruses. You can go after hepatitis C and B, you can go after papillomavirus, you can go after Ebola or COVID, whichever one you would like. And we want to see the results and make sure that everybody, like Sarah, had an issue here downloading the file, right? Maybe you can try again and let us know. But hopefully by next time, we will see different types of pipelines. Now, you can do this kind of phylogenetic analysis on FASTA files, but the FASTA files could also be created from SRA files, the ones that we did before. They just need an additional step of generating a consensus sequence, right? So next time we'll also look at some of the variation of those different pipelines to understand what else we can do with different types of data sets that are available. And then we'll actually look at how some viruses, right? So when we talk about COVID-19, it actually kills people very quickly in a matter of weeks, as opposed to hepatitis B and C, that is a chronic condition over the years, right? As these people reach a certain age, it actually leads to cancer. And then the cause of death isn't the infection itself, it is the cancer, right? So they're viruses that have adapted to their hosts are not as lethal, right? They're very different. They cause mild inflammation, interfere with different signaling, and cause deregulation of this biology that the cell needs to replicate and continue serving its purpose in an organ, in a specific tissue, right? So the modes of action between these viruses is very different, mild and chronic and acute. Like we see in COVID, for example. So those are important elements when we talk about treating those viruses and also understanding how they actually become that way and what is the best approach in terms of vaccines and antiviral drugs that could help prevent them causing some of these severe effects. So I'm going to look forward to seeing you again, talk about the results of these pipelines. In the meantime, if you can continue thinking about your independent project and making sure that you at least go through the step 1, which is to submit your project idea on the form that we showed you that is present in the forum. Okay, and if you have any questions, again, please post them there. Thank you again for joining today. We'll make this recording available for you to review later on. And thank you, Bibsa, for putting that link right there. You can see if you need to put up your questions, you can use that link.